Next, we have Medicenna Therapeutics Corp. It trades on the OTCQX under the symbol MDNAF and on the TSX under the symbol MDNA. It's a clinical stage immunotherapy company focused on developing novel, highly selective versions of IL-2, IL-4, and IL-13 Superkines and first-in-class Empowered Superkines. Happy to welcome President and CEO Fahar Merchant. Welcome to the conference today, Fahar.
Thank you very much for having me.
All right, the floor is yours.
Okay, well, thank you very much. Glad to be here. Thank you for all of you who have joined in this morning or afternoon, wherever you are. Just to let you know that Medicenna is really focused on a series of molecules we call cytokines. And these are engineered cytokines. We've used the technology from Nobel Prize winning work done in the sort of 2018. And these are evolutionary cytokines. And the purpose here is to really generate, we believe, revolutionary medicines for treating not only cancer, but other diseases as well. So just as a good housekeeping, just let you know that I'll be making some forward-looking statements today. So I encourage you to look at our filings and on CEDAR. So starting off with the programs that I'll talk about today is essentially based, as I said, on what we believe are first and best-in-class superkines. So what are superkines? Really, superkines are these engineered cytokines, and cytokines are almost like hormones, really. They really control, they manage, they modulate our immune system. So it really, in the case, let's say, if you had infection, our immune system would be really supercharged. In the case of a situation where we have, say, autoimmune disease, where we will have an immune system that's going to attack our own organs, etc. So having a properly balanced immune system is crucial. But unfortunately, when we have autoimmune diseases or cancer for that matter, that's where our immune system is failing us. And that's where we are developing these superkines to help us treat these patients with either cancer or autoimmune diseases, etc. But today I'll talk really about cancer, and I'll talk about, first and foremost, our lead program. It's called MDNA 11. It's an IL-2 superagonist. This is currently in phase 1/2 clinical trials, and the data look really promising in that in patients who have failed different checkpoint inhibitors, we are seeing response rates of anywhere from 30 to 50% with our MDNA 11 on its own. And then I'll move on briefly to talk about another really exciting space that's becoming, you might have seen a number of news flow just this week. We had Pfizer announce a $6 billion transaction with a Chinese company. And this is where the focus is on generating what we call these bispecific checkpoint inhibitors. These are bispecific anti-PD-1. And I'll talk today about our own bispecific anti-PD-1 called MDNA 113. And we believe that, again, is a first-in-class molecule that we'll talk about later on. The third program that I will not mention much today, it's a program that has generated really promising data in phase 1 and 2 clinical trials. It is a first-in-class IL-4 superkine. This program essentially has an engineered interleukin-4, which carries a very potent toxin. And we've seen really impressive results in patients with very aggressive forms of brain cancer called glioblastoma. And this particular program is now phase 3 ready, with the potential for this program or this particular asset being used for other brain cancers as well, with a very large revenue potential of about $4 billion or so. This program, we are looking to partner this. So overall, what we are from a cash position, we have a healthy cash balance taking us past middle of next year, with several catalysts coming up, most of them before the end of this year. So we believe there's a number of data readouts we'll have. We'll be keen to share those with you in the next 6 months or so. So moving on to our, let's see, where are we? Program here. Just briefly talk to you about the Superkine platform. So just very briefly here, we've taken cytokines interleukin 2, 4, and 13 and really engineered this using this directed evolution approach. And this allows us to generate super agonists or even super antagonists. So in cancer, we need a super agonist. In the case of autoimmune disease, we need a super antagonist. And each of these programs is capable of generating a pipeline in a product. So multiple opportunities with each of these interleukins or superkines we're generating. Moving on to the pipeline that we believe we have a really well-balanced pipeline addressing the three different superkines. But more importantly, we cover early, mid, and late-stage assets. So starting right at the top, I mentioned to you MDNA11. We'll talk more about it. We'll have multiple readouts in the second half of this year. Is currently in phase 2 portion of the clinical trial. We also have with our MDNA11 a separate arm where we are enrolling patients. We are combining MDNA11 with Keytruda. It's a checkpoint inhibitor. It's the world's biggest selling drug. And this is a combination study where we are collaborating with Merck. And then the third program, which I mentioned to you earlier on, which is essentially around bifunctional or bispecific checkpoint inhibitors. This is an anti-PD-1 IL-2 program. And this particular program, we believe, will be ready for IND-enabling studies towards the end of this year, so we can start clinical studies next year. The other programs are earlier stage. One of them is for autoimmune disease, the other one for inflammatory diseases. And as I mentioned earlier on, MDNA55, which is our bizaxofusp molecule, it's an IL-4 fused to a toxin with really promising data in glioblastoma. And this particular program, we're looking to partner this asset. So we'll focus first and foremost on MDNA11. As I said, it's a clinical stage asset. The monotherapy and combination arm, the combination portion being in collaboration with Merck. But before I go and talk to you about MDNA11, let me sort of give you a bit of background on this particular cytokine or interleukin 2. So interleukin 2 was the very first immunotherapy ever approved for treating cancer. And this drug was approved in the 1990s for treating patients with either melanoma or kidney cancer and provided response rates in the 15 to 16% range. But amongst those patients, there was about 5 or 6% of patients that had long-term cures, in the sense that tumors would not come back for 5, 10, or even 15 years. So really exciting particular molecule. But then again, the response rate was not that great. And even more challenging was the fact that this particular molecule was incredibly toxic. So this toxicity meant that patients had to be administered this drug in an intensive care unit because the drug was so potent. It had so many poor side effects, it could kill a patient. And then the second big challenge was this drug did not stay in your bloodstream for long enough, and it needed to be administered every 8 hours for 5 days. So really not something that got traction and was used very infrequently. So what the scientists at Stanford University did was sort of created an engineered version of the IL-2, generating what we call this IL-2 superagonist. And the intent here is really to get much better efficacy, but at the same time have a much better safety profile so that patients can accept and receive this drug in a setting where patients are not in an intensive care unit. So what you can see on the left-hand side, the cartoon basically shows that this molecule has been altered in such a way that it will bind very selectively to immune cells that have this receptor known as the beta-gamma receptor. And that beta-gamma receptor is really responsible for attacking tumor cells. And those are the ones that we want to stimulate. However, the ones that are on the left-hand side that also contain the alpha receptor, those are the ones that will stimulate the immune system. But in addition, they will stimulate immune cells known as Tregs, which will actually protect the tumor. And the other thing is that the binding to the alpha domain also causes undesirable safety profile of the drug. So what we've seen so far with MDNA 11, as I'll explain later on, we're seeing 30 to 50% of patients responding to MDNA 11 on its own. We've seen 100% reduction of tumors in 3 patients. We are also seeing a very desirable safety profile. And really importantly, is that we're not treating the patients 3 times a day, but in fact, once every 2 weeks. So we've got a molecule that's got unique pharmacology, balancing safety and efficacy. So how have we sort of achieved that? And essentially what you see on this cartoon is that on the left-hand side is the engineered IL-2. We have inserted a couple of mutations where the molecule is no longer able to bind to the alpha receptor. So that will mean that this particular IL-2 will not stimulate the cancer-protecting T regulatory cells and also will avoid the safety issues. The second thing we did was insert 5 mutations to substantially enhance binding to the beta receptor. And by doing that, we are able to stimulate the cancer-fighting immune cells. But also what we found is that we are able to stimulate what we call memory immune cells. And these memory immune cells stay in our bloodstream for a very long time. So if the tumor comes back, the immune cells are ready to attack the tumor again. And then finally, what we did was we linked our IL-2 to albumin. So albumin is a large molecule, means that it will not live or it will not clear your kidneys very quickly, and therefore the drug stays in your bloodstream much, much longer. And this way, we are able to treat patients once every 2 weeks. The other big advantage about albumin is that it tends to accumulate in the tumor, but even more importantly, it accumulates in what we call the tumor-draining lymph nodes. And this is where the immune system gets trained to fight cancer. So overall, we've sort of incorporated in MDNA 11 a number of key features that make the drug more efficacious, safer, and generate immune system or immune responses that are long-term. So the trial that we are conducting right now is what we call the Ability-1 clinical trial. This is a phase 1/2 clinical trial. We finished the phase 1 portion of the study. So what we did was initially conducted the phase 1 with MDNA 11 alone, and then subsequently conducted a phase 1 study with MDNA 11 in combination with Merck's drug Keytruda. And in both cases, we identified the 90 microgram per kilogram dose as being the ideal dose that we are now in the phase 2 portion, where we're using the same dose of MDNA 11, either on its own or in combination with Keytruda, to treat a number of different tumor types. So on the right-hand side, you can see a list of different tumors. The first one at the top is melanoma. The second one is MSI high DMMR tumors. The third one, TMB high. And the fourth one, in the case of monotherapy, we are treating patients with tumors that are evolved as a result of viral infection. And then in the combination setting, looking at a combination and sort of looking at specifically gynecological tumors. Now, the reason we picked these tumors, especially the first 3 tumor types, is that each one of them would be patients who have failed checkpoint inhibitors. So currently approved checkpoints these patients are not responding to those therapies. And second of all, there is a pathway for accelerated approval, meaning that through a phase 2 clinical trial, a single arm could generate a sort of a approval with either on its own or combination for these 3 different tumor types. So that's where we know what the benchmarks are. For melanoma, for instance, if we get response rates in the 30% range, that would be sufficient to get to that threshold. With the MSI high and TMB high, so these are biomarker driven. So essentially, it's not a particular organ that's has this particular tumor type. There's sort of dozens of different solid tumors that have this biomarker known as either MSI high or TMB high, and therefore you can treat so many different tumor types. But these patients would have failed checkpoint inhibitors, and here the threshold would be to secure response rates in the 20% range. So looking at safety, we're seeing a really desirable safety profile. We did not see even as high as 120 micrograms per kilogram. We did not see any dose-limiting toxicities at any of the cohorts, either on its own monotherapy or in combination with a checkpoint inhibitor. And by and large, as you can see, the adverse events were generally of grade 1 and 2. So more than 90% of the adverse events were grade 1 and 2. There were a handful of grade 3, 4 events. And we can see that that was the case even in the combination setting as well. So we're not seeing any new adverse events in the combination setting. And whatever we're seeing in the monotherapy is what we would normally expect with IL-2 molecules. But the important thing here is that the safety profile, most of the adverse events are low to moderate, but more importantly, they are of short duration. So these adverse events go away in 24 to 48 hours. The other thing what we found is that as we had designed the molecule, what we end up seeing is really exactly what we were expecting, is that we were preferentially stimulating the cancer-fighting immune cells, namely the CD8 T cells, and you can see the expansion of CD8 T cells quite dramatic, whereas the Treg, which is sort of the gray, virtually does not even show on this particular slide, showing that we are really boosting the cancer-fighting immune cells more effectively than the Tregs. So it's not only the cancer-fighting immune cells that we are stimulating, it's not only the quantity of these immune cells that we're boosting, but it's the quality of these immune cells. And here we are seeing that when we look at the very specific subsets of these immune cells known as CD8 T cells, which are the ones that are the most prolific, the most potent cancer-fighting immune cells, we see a dramatic increase when we compare it to baseline to day 8, with each of these immune cells showing that these are much better cancer-fighting immune cells. They have these certain markers known as DNAM or TCF. These means that these immune cells stay in our system for a very prolonged time. And we also see the same thing with memory CD8 T cells, meaning that these immune cells will allow patients to have a treatment and receive therapy, but the durability will be prolonged, so the tumor does not come back very quickly, which is what we see in our clinical trial. So based on that, if we look at the data we've generated so far with MDNA11, this is MDNA11 on its own. This is in patients who have failed at least one, if not two, in some cases, 3 different checkpoint inhibitor therapies. These patients, you can see, have prolonged and durable clinical benefit. And this is really important, particularly if you look at the very top, the very first patient there. This is a patient with pancreatic cancer. And this patient initially had surgery, subsequently received a cocktail of 4 different chemotherapy drugs. The tumor continued to advance and eventually metastasize and spread to the liver. The patient then received another combination of 2 different chemotherapies the patient was not able to tolerate, and then finally received Keytruda. And that Keytruda itself continued to show that the patient was progressing. The patient entered our clinical trial, received MDNA11. By the first scan, we saw the tumor was stabilized. By the second, third, fourth scans, we can see the tumor shrinking. And eventually the tumor shrank to about 70% or so. Patient went away on vacation, came back after 8 weeks of holidays, and unfortunately there was a new tumor that showed up. And the patient was briefly treated with radiation for just 1 cycle and started MDNA11 again. And what you see is that that patient's tumor completely disappeared, not only the ones that were in the liver, but even a new one. And that patient has been off therapy now for about a year and a half, and there is no sign of the tumor coming back, which is consistent with what I mentioned, this potential, the curative capability of a drug such as MDNA11. We're seeing the same thing with a number of other patients. And hopefully, as you can see from this particular slide, we are seeing very impressive, in fact, remarkable responses in patients ranging anywhere from 30% to 50%. And in some cases, a couple of cases, as you can see, the tumors have completely disappeared. So really encouraging data we've seen so far in patients with MSI high tumors, for instance. We are seeing a response rate of 50%, which is really impressive amongst patients with secondary resistance to immune checkpoint inhibitors. We're seeing response rates of about 33%. And amongst all those patients who are eligible for the phase 2 portion of the trial, we're seeing a response rate of 40%. So each cases, we're seeing really promising results. And then when we look at the combination setting, again, we're seeing response rates of 30 to 50% in different tumor types. So these are patients with gynecological tumors. We're seeing, for instance, patients with endometrial cancer, where these patients would not normally respond to a checkpoint inhibitor. We're seeing a 50% response rate. In a patient with anal squamous cell carcinoma, where checkpoint inhibitors really don't show much responses, it's usually a 5 to 6% response rate. We've already seen that one patient have a complete response after the first scan. And then we're also seeing patients with other tumor types, including colorectal cancer, melanoma, etc. We're seeing response rates of about 36%. So each one of those are really exciting data sets. We're seeing responses in tumors that normally don't respond to checkpoint inhibitors, and we're seeing these responses to be durable. So overall, the key highlights here that MDNA11, either on its own or in combination, is generating response rates in the 30 to 50% range. We're seeing really promising safety and efficacy in the combination setting. And we are able to treat patients once every 2 weeks, which is so much more better than treating patients 3 times a day. So overall, we continue to advance this program. We'll have more data coming up in the next 2 quarters, and these will be key value inflection milestones for us in order to prepare us for phase 2 registration trial. Moving on to this other new program that I briefly mentioned to you, this is the bispecific checkpoint inhibitor, MDNA113. So what is this excitement around? And as you can see from this particular slide, anti-PD-1 inhibitors, such as Keytruda or Opdivo, are today the best-selling drugs. Keytruda particularly is the number 1 selling drug. In 2024, annual sales were $30 billion. And these drugs are expecting their patents to expire in 2028. And that includes Opdivo with sales of about $12 billion. The thing is this, that as this patent cliff is just around the corner, there's an attempt to create these bispecifics where these checkpoint inhibitors are fused to other candidate molecules such as VEGF, and we are working with an IL-2. And you've seen multiple transactions. Each of these are either in early stage or mid-stage clinical trials. You see transactions of Merck, Summit, BioNTech, etc. Each one of those, you can see these are multi-billion-dollar transactions. And just this week, we had Pfizer enter into a deal with 3S Bio from China in a $6 billion transaction with $1.25 billion upfront for their bispecific. What we're doing with our drug is quite different. In fact, we are taking an anti-PD-1, which is already approved in the marketplace. So we are not experimenting with new anti-PD-1s. The second thing is we fused it. That green dot that you can see, or the green circle that you see, is our IL-2, which has already shown single agent activity on its own. And then the pink squares that you see is our IL-13. And this is really unique in the sense that it sort of allows the drug to be targeted to the tumor because the IL-13 will bind to a certain receptor on cancer cells, and it only bind there and not healthy cells. And once it's located there, the IL-13 is cleaved off, the drug is activated, and you switch on the drug right at the tumor site. So you can see what's happening on the left-hand side. The molecule, the IL-2, is masked. And as it gets into the tumor site, it is unmasked, gets activated. And that's where we find the key major advantages of our drug with MDNA 113 and other bispecifics that we are working on, is that we have a number of key advantages compared to other bispecific checkpoint inhibitors. So the reason we are pursuing with the IL-13 version is that every year, about 2 million patients are diagnosed with tumors that express this particular antigen known as IL-13 receptor alpha 2. And you can see that this is an opportunity for us to really treat patients with these, what we call cold tumors, where checkpoints on their own do not work, and for us to address this huge unmet market. So just for those who are not familiar with the checkpoint space, we know one thing is that with checkpoint inhibitors, about 30% of the patients will benefit, the remaining 70% do not. And the majority of these are these ones that you see here with the IL-13 receptor alpha 2 tumors. So that's where we are. We have an exciting set of data coming up in the coming quarters. As a company, we are well capitalized. We have sufficient cash to take us into Q3 next year. We are headquartered in Toronto, about 90 million market cap, 30 million in cash. We generally spend about 4 to 5 million every quarter, with insider ownership of about 22%. We are covered by various analysts, 2 of them in Canada, 2 of them in the US. And in addition to the key advantages that we see with our programs, is that the multiple milestones expected over the next 2 quarters. We'll have full set of expansion data in the monotherapy, which is MDNA11 on its own. We'll have completed the entire enrollment in the clinical trial, and we'll have top-line data of MDNA11 in combination with Keytruda. In addition, we'll be looking to partner our bizaxofusp, the GBM program, and finally, we'll be ready with MDNA 113 for IND-enabling studies before the end of this year. So multiple milestones coming up over the next 2 quarters, and I'll stop there. Thank you.
Wonderful. Thank you, Fahar. A few questions for you with the time we have left. First, the AACR is one of the biggest cancer conferences of the year. It just took place a few weeks ago. So talk a little bit about that. What was the general reception like as Medicenna's posters presentations went off at that conference?
Yeah, it was just quite amazing. In fact, during the poster session, which lasts about 3 hours, we were surprised that we didn't get a chance to even move away from our posters, with 2 of us standing and answering questions. So it was pretty much packed around our poster. So it was really busy. On the second day, on the last day of the conference, we actually had security, ask us to leave because we had so many interested parties wishing to talk to us. We had individuals from sort of the large, big pharma come and meet with us, really excited about the data. So really encouraging. We presented data on both MDNA11, but also our bispecific MDNA 113, where there was a lot of excitement. So yes, very well received, and we are keen to share more data in the coming quarters.
Fantastic. All right, so give our audience about 30 seconds of closing remarks. We are out of time. We've got lots of questions for you. We'll send, but 30 seconds of closing remarks for you, for our viewers today.
Yes, so just to let you know that Medicenna has a really exciting pipeline around the immune interleukin space and building a pipeline around bispecifics, checkpoint inhibitors. And we have multiple data readouts coming up, with the intent that these readouts will address the huge unmet needs for patients who do not have therapeutic options, including checkpoint inhibitors, where these patients fail those therapies. And we believe MDNA11, either on its own or in combination is already demonstrating response rates of 30 to 50%, with some patients having really long-term benefit. So look forward to sharing more data, and we're excited about the data set, and hopefully it puts us in a position where we are open to potentially partnerships, collaborations, etc., in the next 2 quarters. So looking forward to sharing more information with you in the coming year.
Wonderful, Fahar. Thank you for your time today, and we hope to see you again real soon.
Thank you. Take care. Bye-bye.
All right, everyone, we'll be right back.